With that, it's my distinct pleasure to introduce the Pierluigi Gambetti Honorary Lecturer, Simon Mead. I do want to, every time I say this, someone comes out afterwards. Pierluigi is still alive. He's still with us. Suzanne and David spent some time with us. He's doing well. It's just he's such a special person. We had to name the lecture after him. He was the CJD Foundation's first medical director and the first director of the Prion Center. Following in his footsteps for today's lecture, Dr. Simon Mead is professor of neurology at UC College London. He works in their Prion lab. He has been very implemental in many things in the field, including genetics, but also developing good outcome measures for things like clinical trials in this field. So Simon, thank you very much for agreeing to come. We appreciate it.
Thanks so much. It's a really great privilege to give this lecture. But I have to say, if I'd known dogs were invited, I would have brought my own and would have shown you some photos, but you'll have to follow me on Twitter to see pictures of Betty. But look, I think it's a really, really super conference. And I'm afraid I haven't been as many times as you, Brian, but I do intend to make it more regular in the future. I think probably it's the only conference that really draws together people touched by the disease, physicians and scientists, in a really special way. And so, yeah, really pleased to present my work. As Brian said, I'm a clinical neurologist. I work in London at the National Prion Clinic and run a genetics research lab in the associated MRC Prion unit at UCL. And I'm going to give you a flavor of the work I do now. Oh, actually, I don't know how to move the slides. Is this a slide advance? Great. I'm back. Super. And it points as well. Great. So for those people that weren't present at some of the introductory talks yesterday, I'm going to just cover some of the brief biological mechanisms of prion diseases, and then I'm going to give you three kind of snapshots of the kind of work I do. I'm going to talk about the inherited prion diseases first, as I'm talking about genetics. But in particular, how can we use looking at this particular type of the disease to understand the really early stages of disease? We all want to get to a complete cure for the disease. That's the idea, or complete halting of the disease. But I think it's obvious to many of you that have looked after patients with sporadic CJD, the disease is just diagnosed too late in order to make that effective. So understanding the early stages, even predicting it before it starts, could be a critical step. I'm going to tell you some other varied stories as well about work with acquired prion diseases, Kuru, and how we've looked at a distant population that have evolved resistance to the disease and how we might exploit that. And then finally, I'm going to talk about a big part of my work, which is collecting large numbers of samples from you all, from families, from patients with sporadic CJD, to try and understand risk factors, new targets, to try and develop the basis for future treatments. I can talk about this for a long time, so please do stop me if I'm overrunning. I can see the time there. I started just perhaps a little bit before 9 o'clock, so I'll try and make sure I finish on time. So let's cover some of these basics I referred to. There are three different ways in which you can get prion diseases. The kind of archetypal, the basic form is sporadic CJD that seems to come out of the blue without any obvious explanation. That's in the vast majority of people, 85%. I'm going to talk in the next section about inherited prion diseases. Families are affecting about 10 to 15% of the total. It does vary a lot between different parts of the world. And then there's these rare but important for public health purposes, these acquired forms, which are obviously also very important in the veterinary diseases. So just this recap of a prion protein, the gene, and prions themselves. So DNA is this encyclopedic molecule that we have in cells of the body that contains an instruction manual to make all of our proteins that do the jobs in our bodies. It's a code that has 3 billion letters, 4 different letters, 3 billion long, and that encodes all of the proteins. But how a cell makes those proteins is by sending little parcels of message outside the nucleus where the DNA is contained for how to make an individual protein, and that's done as RNA. Proteins themselves are made of chains of amino acids, like beads on a necklace. The prion protein itself has 253 different amino acids, and that has to be folded in a particular way in order for prion protein to do its job. I kind of conceptualize prion protein as like a kind of globular little bit with a tail that doesn't have a clear structure. And then that has to be moved in the cell to the right place in order for it to work, and some of the amino acids are cleaved off, sugars are attached. That's the basic biology of how prion protein, the normal prion protein, is made. We've known the structure of the normal prion protein for a long time, maybe 30 years. That's shown on the left-hand side. And as Joel and Brian described yesterday, it's predominantly formed of these helices, 3 different helices that are stuck together, and it's bound to the cell surface. But until recently, we've only had a schematic mechanism of how prions actually form. And in this concept, you have to imagine a seed or a fiber formed of a misfolded form of this protein. These proteins are stuck together, and they form a rod-like structure. And that rod has this odd property that it can bind the normal form of the prion protein, and it can stamp its shape onto that normal form, turning it into the abnormal form, and therefore growing the rod. And then that rod can fragment, and those little fragments move around cells and move between cells, and that's how the disease spreads and grows like an infectious disease. But curiously, there's not just one way in which the protein can misfold. There are different ways it can misfold, and these different shapes are propagated through this templating, this stamping mechanism, and these cause the strains that we're familiar with, like the differences between different types of sporadic CJD and variant CJD. So I mentioned until recently. Well, until recently, we've begun to develop pictures of what prions actually look like. And there's a variety of images I'm showing on this picture. Some like photographs taken by electron microscopes, but other kind of computational images of what prions actually look like. And they seem to be kind of rather stiff, straight, twisted rods, microscopic, really tiny, tiny things, sometimes found on their own, sometimes paired with other rods. And cross-sectionally, the protein looks almost like a pulled paperclip or hairpin in a kind of V-shaped structure, completely different in shape to the normal form. And it's that transition that is critical to prions being formed in the body. Okay, so that's the section on the basic biology of prions. Now on to my first story, which is about how we can use inherited prion diseases to study the early stages of disease. So I'm showing two really large pedigrees here that are of individuals that have developed inherited prion disease from the UK. These pedigrees don't show all individuals, they just show the connections between people who've been affected, going back over many, many generations. So as many of you probably know, the inherited prion diseases are passed on, typically affecting just half of each generation. But these pedigrees would be far too large to show in their entirety. The mutations passed on in these families were some of the first mutations that were discovered in the prion protein gene going back into 1989. And since I joined the prion field in the late 1990s, I've been working with large families like this and others with different mutations to do clinical research work. So here in this image, I've taken the prion protein and I've kind of straightened it out and illustrate some of the different types of inherited prion disease that are common in the UK and that we've studied in this work called the National Prion Monitoring Cohort Study, which I'm going to tell you a little bit more about. So perhaps it makes sense to start with this one, E200K, which is the most common mutation worldwide. So in recent years, we've seen 23 patients with this condition, and we study people that have had a predictive gene test that know they carry this mutation, they're likely to develop this disease later in life. And we've followed up 35 people like that, 6 of whom suddenly converted to disease during the study. So this disease typically starts in late middle age or early old age, but it's a fast disorder. It looks just like sporadic CJD, really, with thinking skills affected, balance, jerky muscle movements, and often it's misdiagnosed as sporadic CJD in the first instance. Probably the most common one that we see in the UK, though, is quite different. It's associated with a P102L mutation. So recently we've seen over 40-odd patients with this. And we follow up in the cohort 31 healthy carriers, 11 of whom suddenly have converted to the disease. This also starts in late middle age, but lasts for about 5 years, and normally starts with a balance problem, difficulty walking, pain in the legs, bladder problems, and only later problems with thinking skills, sometimes with psychiatric problems. The UK, I think, is quite unusual in that we see a lot of patients with this mutation called 6-OPRY, where there's extra amino acids in the prion protein. And we've seen 30 or 40 patients over recent years with this mutation. This is quite unusual in how early it starts. Sometimes it starts with the first symptoms in teenagers, often in young adults. And it's distinct in that it affects behavior and personality, and only very much later affects movements like we see in other prion diseases. The duration is also much slower, over 10 or 15 years. We do have some families with the D178N mutation, which is common in Germany and other parts of the world, but fewer patients that we followed up in the cohort study, and 8 healthy carriers. This condition is fast and also affects the autonomic nervous system, can affect blood pressure, pulse, temperature, and sleep problems. And finally, this unusual mutation that actually truncates the protein, it kind of cleaves off the latter half of the protein, and we've seen a few families with this and related mutations. This can also start very early in life and unusually has a non-neurological presentation, can present with sickness and diarrhea, abdominal pains, and nerve problems due to deposition of this free-floating prion protein in small nerves of the body. So there are several others as well that are as common as 163X or less common that collectively we see in the cohort study. The cohort study involves sporadic CJD patients as well. I'm not going to talk about that today. That's about 1,200 participants now. But in particular, the focus here is on the 100 people that we follow up that carry gene mutations. And this work now is led, initially started by me, but now is led by this super doctor, Dr. Mock in our clinic. So out of those 100 we're following now, there's 20 in total that have developed symptoms within the study. So the typical pattern here, and this chart here is showing kind of a summary of how we've followed people up. So each line is an individual, and each dot on that line is when we've seen them and taken samples, or spinal fluid samples, blood samples. And although you can't see it so clearly in this projection, there's a dotted line there. Sadly, when people cross that dotted line, that's when they've developed symptoms of the disease and been diagnosed with it. And of course, whilst that's very sad, and certainly I've become very close to some people that have contributed so much to the research studies, seeing them every year for maybe 10 or 15 years. It's obviously a tragic event developing the disease, but it's also a fascinating contribution to research to be able to look back at those visits and the samples that they've given before they developed the disease and see whether there was anything we could see in there that might have foretold what was going to happen. So it's not just blood samples, though. We look at brainwave tests, brain scans, neuropsychological assessments, the clinical examination. We feed back the results collectively, not individually, so we don't feed back these research results to each individual, but as a group. And we also hope to contribute to the well-being of people as well, and we're looking to formalize that through workshops called acceptance and commitment therapy workshops in the near future. But what I wanted to talk about was the results that we're getting from looking at the various fluid markers prior to the onset of disease. And I think in order to think about this, you have to understand how different the different mutations are and split them up into either fast disease or slow disease. So you take the fast disease, the best example being the E200K mutation. When we look back at the spinal fluid tests in the years leading up to the start of the disease, and we do the RT quick test looking at this amplification of the prions from the spinal fluid using synthetic protein, we see that everyone we've seen in recent years has had abnormalities in this test in the years leading up. Sometimes just a few years, sometimes over 5 years. But we haven't seen a case now of E200K that has developed the disease without having had a positive RT quick test in the spinal fluid in the years leading up to it. And that's been the case for nearly 10 years now. If you take the more slowly progressive inherited prion diseases, the one that we've seen most experience of being the P102L, you see a different picture. We don't have an RT quick test that works well in these patients, so we can't do the same kind of thing. I think that's an important gap that we have. But what we have been doing here is looking in blood at proteins from brain cells, either neurons or supportive cells of the brain, that when they're damaged, they leak into the spinal fluid or surrounding fluids. That then leaks out into the blood. And using very sensitive assays, we can detect the leakage of those proteins. And I think, although it's very difficult to predict in individuals with confidence about what's going to happen to them. I think collectively, when you look at groups, you can see this rising pattern of increasing damage to nerve cells. This years to onset 0 then is the time at which the disease is diagnosed, and this grey area is a time at which we feel there's a clear pattern of rising brain proteins that we can detect in blood. So we'll conclude this little one of the triptych of examples of how genetics might contribute to the disease. We've worked with people, I've worked with families over 20 years now, 20 years plus, to try and give insights into these early pre-symptomatic stages of disease. This is long-term and difficult work on both sides. This is really altruistic time that's given by people traveling to London for these series of tests. It also reminds about the disease, which I know carries a psychological burden. But I think it's the only way that we get insight into these pre-symptomatic or early stages. And there's two conclusions, really. For the fast mutations, we're detecting abnormal RT quick in the spinal fluid in the years leading up to the disease. In the slow, we see a progressive rise in brain proteins that leak into the blood, which is clear in retrospect in groups of people. It's not really so clear in an individual to give confidence that you can say there's a strong expectation something going to happen. But I think there's a reasonable chance that we will get there. I should say I've presented my own work here, and I'm aware there's a number of groups around the world that are doing similar work, some of which has considerable overlap with the work I presented, in particular to Flag Sonia and Steve Arnold's work, Inga Zer's work at Göttingen. Michael Geschwind is doing similar work, and neurologists in Israel too. So in the wider context, I spend a lot of time on the UK's National Prion Monitoring Cohort Study. It's the ambition more widely to inform the design of clinical trials and targeting of treatments by diagnosing the disease as early as possible. And I hope also, through studying patients with sporadic CJD through the disease, we can find a way to design drug trials that don't need to give placebo tablets. That's an ambition over coming years anyway. Okay, that's the first 9:15. I'm doing okay. I'm on time to finish my 3 stories. So the next story is something completely different, okay? So I'm going to talk about genetic evolution of populations to protect themselves against prions. And here we need to travel in our minds right across the world to Papua New Guinea and to the eastern highlands of Papua New Guinea. Go back to the 1950s. This is a particularly remote and inaccessible part of the world, but especially so in the 1950s, where there was very little contact between people that lived in this area and the outside world. In fact, there are large populations that live in this region of the Eastern Highlands province, a dense tapestry of populations with fascinating and diverse cultures, languages, but one that particularly attracted attention of the rest of the world were the Foray people that in the 1950s were found to be suffering from an epidemic disease, an epidemic disease that started with balance problems but very rapidly developed into dementia and was devastating the communities there, particularly women, but also children. And this was called Kuru. And epidemiologists working in the region realized in a few years that this disease was a brain disease that was transmitted by cannibalism. And cannibalism was a behavior within the Foray people as a way to respect the dead. And when somebody died, in order to respect their soul, they had a ritual whereby the body was broken up and different parts of that body went to, in a ritualistic way, to kith and kin. And in particular, the prion-infected brain tissue was distributed to women, adult women in the community and the children that lived with them. And that resulted in the disease. Sometimes after short incubation times, the disease affected children as young as 4, but alternatively really long too, in that we've seen Kuru patients that have developed the disease this century, even though the cannibalistic practices were outlawed rapidly by the Australian colonial administration and certainly stopped by 1960. This is typical Foray terrain. It's quite tropical jungle, quite mountainous, impassable rivers, quite a few infectious diseases around, and typically Foray hamlets would be on the top of the hills where there were less insects. This is a typical Foray hamlet, and this is a typical Foray research laboratory where where our research nurse, Jerome Whitfield, and a local team of P&G researchers set up to visit people in the foray, collect samples, process them. So this was funded by the Medical Research Council because in the 1990s we realized that there were parallels between Kuru and what the UK faced with an epidemic of BSE that had crossed a species barrier, was started to affect humans as variant CJD. This was an orally transmitted prion epidemic. The only previous example was the devastating epidemic that affected the foray. And we thought there was something to be done by jointly researching this. So in this research lab, there's a centrifuge that's hand-powered, and there's a solar-powered fridge and freezer. We have to say we've complained to the MRC on a few occasions that the laboratory wasn't sufficiently audited by their health and safety teams, but that's when they were onerously investigating the London labs. Okay, so on to the epidemiology. So I'm showing some maps here, and these maps show all the villages that were carefully documented by Michael Alpers and his epidemiology team, mostly P&G researchers doing field work and going out to see people that might have Kuru, and documented every village where there was ever a single case of Kuru. So this shows the South Foray villages in some detail. And you can see the different areas of grey and white. And these reflect different regions of increasing Kuru prevalence. So there were some villages that just had the occasional case of Kuru, but there was a central region where Kuru was especially prevalent. Blood samples were taken from these communities to help with research. And in particular, what we were interested in was this myth that if you attended a mortuary feast, if you attended one of these ritualistic ways to respect the dead, that you would die from Kuru if you had eaten infected brain tissue. But in fact, this was a myth. It was not correct, because Jerome and his field team were able to interview hundreds of people that had in fact attended these mortuary feasts and in fact survived, had not developed Kuru. And so our question was, why was that? Was there something? We've all seen the disaster movies, haven't you? You know, what's the standard plot? It's like, oh well, the world's going to end, everyone's infected with a virus, and then somebody survives, and scientists study them and then find what it was, and then inject everyone else. It was a similar kind of principle here. So these people had survived, they'd eaten Kuru-infected brain tissue, that survived for more than 50 years and more. You know, what was the reason? So we first of all started to look for genetic explanations. So we sequenced the prion protein gene. We determined all the variation within it in people that had survived. And the initial striking finding was we found something completely new that we had not seen before. And that was a variation at position 127 of the gene. And then when we looked where these people resided, it turned out they were entirely co-localized with the center of the Kuru epidemic. And so our initial thoughts when we looked at this was, well, we thought, well, maybe this was the trigger for Kuru. Maybe this was an inherited prion disease, like the ones I described in the first section of my talk. And maybe that was the trigger. One of those brains was eaten, and that's what sparked the whole thing off. But then when we looked at it in more detail, when we did the statistics on it, and looked at younger people, survivors, and also looked at this polymorphism that you will have heard of from Joel and others at position 129, which is also highly prevalent. These pie charts here, these pie charts show people that we sequenced for this genetic polymorphism at 127 and 129. And you can see, if you go outside the regions of high Kuru and more distant as well, most people are homozygous, meaning they have just the same prion protein gene. They have no variation in it. But if you go into the center of the Kuru area, the vast majority of people that survived Kuru have either codon 129 variation or codon 127 variation. And we showed genetically as well that these polymorphisms had increased in frequency, most likely in response to the Kuru epidemic. So in fact, the foray population were evolving to protect themselves against the disease. So we had this idea then that this novel thing that we'd found at position 127 was something protective. So how did we go on to test that? And that was done using animals, research led by Emmanuel Asante, who's now left the unit. But he made a transgenic mouse that has a human gene that carries this 127V variant. And when we made these mice, we tried to infect them with human prions, either Kuru, sporadic CJD, variant CJD, and these mice were pretty much resistant to everything we threw at them. So there was some very low-grade replication of variant CJD prions that had never affected the foray, but other types of prion they were completely resistant to. So what have we been doing since then? Well, the grand scheme here is to use modern genetic techniques to find evidence of other genes, not just the prion gene, but what in other ways had the 4A people evolved resistance, and whether this can generate new targets, because the 4A, say, for example, may have taken out a particular gene that's vital to help prion protein develop into prion diseases. But early on in this work, we realized that actually we knew pretty much nothing about the population genetics of this region, and it was impossible to do these evolutionary studies without investigating that further. So for the last few years, we've been doing a background population genetic study of the region, studying these populations so that we can use tests of evolution. It's remarkable what we found. The diversity in this region of the Eastern Highlands province is remarkable. It's an area about the size, what I say of Jamaica is the comparison that I have. I also looked up US states. It's the same size as the US state of Delaware, which I think is a very small US state. But there's more diversity within that region than there is in all of Europe. In terms of in genetic terms. So it gives some capacity for the possibility of variation in other genes that will have protected the 4A. So in this population genetic research, we've described for the first time the ancestry of these populations and information about how people migrate between the different linguistic groups and how the 4A population had to respond to Kuru by attracting brides from long distances in order to maintain the population after some villages became almost devoid of women altogether. And that's, if you're interested in those stories, there's an accessible article written up in The Guardian just a few months ago that describes this work. So we'll conclude this section, one more section to go. So we worked with elderly survivors of Kuru to understand how they were resistant to disease. Nearly all survivors carry variation in the prion protein gene, a novel variation at this position 127, but also this common polymorphism at 129. We made mice that express the variant and showed that they are almost completely resistant to disease. And in ongoing work, we're looking to find other genes that might have evolved resistance to Kuru. As for treatments and possible angles that way, a question that arises is there any way to deliver the protection that 127 affords the 4A to patients or people that might be at risk of prion disease? And certainly there are avenues that we can explore there. These experiments, Tom Cunningham, that's here as mouse geneticist that works in the unit, is exploring delivering prion protein with 127V into brains of mice to see whether or not that, as a form of treatment, a virus-delivered treatment, might have some merit. And if you're interested in talking about that further, I'm sure, Tom, you'd be happy to do so. Last section. Okay, I want to talk about the most common form of prion disease here that's probably affected or touched most of the people in the audience, the sporadic CJD form. So one of the things that my lab leads on is trying to detect other genes that might be responsible for risk of developing sporadic CJD. And this takes advantage of the diversity that we have in our population. So if you look around the audience, you can see, well, we're all different. Okay, a lot of that is age, sex, and there's superficial differences like skin color, height, eye color, and these are genetically determined, and the same applies to molecules in our body and our organs. And some of those differences might well confer altered risk of developing sporadic CJD. Now, I'm not talking about deterministic risk here, like the families I showed, where the gene is passed down from one to the other, but maybe subtly different risk. So whereas Brian talks about 1 in 6,000, maybe for some people it's more than that. Maybe it's 1 in 3,000, 1 in 4,000, and others are never going to get sporadic CJD because they're resistant. So a way we research this is by collecting together really large samples of patients that have had the disease and comparing them to the healthy population and kind of looking at their genomes. So we published work on this. This chart here shows a kind of map of the genome and where this risk lies, and we were pleased to see that the prion protein gene, its code is PRNP, kind of stands out there. This is called a Manhattan plot because you see these kind of towers of statistical association, and the prion protein gene was the top of the list. But we also saw some other signals coming up as well. There's this gene syntaxin 6 here, which Tom Cunningham's going to talk about a little later. And another gene here, an enzyme, GAL3ST1, that's an enzyme that adds a sulfur group to lipids. So these came out of the blue. We had no reason to think that these were risk factors for sporadic CJD, and it opens up completely new avenues of research. But I presented this work to the scientific literature a few years ago, but since then I wanted to talk about what we've been doing more recently, and there's been some super computational models developed that allow you to take a genome data and use that to model the levels of RNA and the levels of protein in different parts of the body. In particular, here I'm going to present work that's looked at modeling protein levels in the brain. So you can take genetic data from an individual and predict from that how much of all different proteins are going to be made in different parts of the brain. And in this statistical chart, I show you across the genome or show you across the proteome levels of different protein in brain region based on modeling from two different reference groups. You notice the prion protein gene doesn't show up here. So it seems as though genetically determined protein levels of the prion protein itself don't determine risk of sporadic CJD, but the other proteins show up, and the syntaxin 6 again shows up as the top hit in this study. Protein levels of syntaxin 6, increased protein levels seem to give you increased risk of sporadic CJD. But there are other proteins too. This one, MANF, came up, and this one, PDIA4, which was nearly a hit in our genome-wide association study. Now, the MANF is significant in this resource, not in this one, but is close, so it's rather convincing. How do we put it all together? So, well, the main risk, the main event in sporadic CJD is a change in shape of prion protein, as we've described. There's no doubt about that. Nothing in this work is going to confound that, but it seems we're finding evidence of other partners. I'm going to just give you an overview of what I think are some of the main players that are derived from this kind of work. So this enzyme GAL3SD1 adds a sulfur group to lipids, and that sulfur group makes them like water at one end. We think they may cluster together, and they may well be potentially a cofactor that is involved in prion formation. The syntaxin 6 moves things around a cell, so it takes things in vesicles and encourages those vesicles to bind with other parts of the cell. So we think this, what we call intracellular trafficking, so moving prions around cells, is also a crucial risk factor. And the other thing that's flagged up is the cell's response to stress, sometimes referred to as the unfolded protein response as well. So we found genes there. There's other evidence that that's important in prion disease risk. And finally, the other surprise, the real surprise for me, was a cell type that came out. Syntaxin 6 increased levels and the GAL3SD1 have quite localized expression in the brain to a supporting cell called an oligodendrocyte that makes the fatty substance that surrounds axons that allow nerve cells to communicate with one another. Now, previously, there was nothing really known about these cells in prion disease. There was no real evidence they were main players, but it's interesting that the two big risk factors that we found in sporadic CJD both implicate that particular cell type. So I'll finish off. I think I have gone over a little bit. Apologies for that. I've told you three stories, one from inherited prion disease, one from acquired, one from sporadic, about the impact of genetic research in our field. I've told you about long-term work that we do with people that carry gene mutations in order to study the early stages of disease and find markers, biomarkers we call them, that might predict onset. I've talked about work with the foray people to find out how that population evolved to be resistant to an epidemic prion disease. And I've told you about working with large numbers of sporadic CJD patients from all over the world, together with computational biologists, to try and discover new targets. And I think it is important to discover new targets, the more the better. But it's important for me to emphasize in wrapping up that I don't think anything in this work does anything to go against the idea that, of course, the prion protein gene and the prion protein itself is our number one target. It's such an exciting time to work on CJD as technologies that target prion protein, our number one target, enter the clinic. And it's by far the top target. Although I have to say, some of you may know that our own therapeutic development and immunotherapy, an antibody called PRN100, has not moved forward. We treated 6 patients with this a few years ago, but due to lack of funds, we have not been able to progress this to a pivotal clinical trial. I'm afraid it just remains stalled. Most important in closing, I'd like to thank you, patients, healthy people at risk in the UK, PNG, and worldwide for participation and collaboration, as none of this work I've presented here would be possible without that generous time. Also to acknowledge some co-workers. This is the National Prion Clinic team. So we have a mixture. John Collins is the director of the unit. And I'm the clinical lead of the clinic, and we have doctors, neuropsychologists, nurses, administrators, and this is where we're based. The clinic is based in the same building as the laboratory, so a lot of the doctors we work with, they do a PhD in the lab, and there's a really great interaction between clinicians and research scientists in the same place. If you want to see photos of my dog, that's my Twitter handle. Apologies for people that go there expecting to hear about science. In terms of the genome-wide association studies, really I have to thank the entire Prion research community, people that see CJD patients, and all of you families who've donated DNA for research. It's probably gone into these studies, so it's a collective thank you. But in terms of the proteome-wide association study and transcription-wide, I have to thank Fahi Kukukali and Crystal Sleegers, who are excellent computational biologists based in Belgium. And for Papua New Guinea, of course, most importantly, Papua New Guinea researchers, and I can't show them all, and I'm showing there Willie Pomat, who's the current head of the director of the PNG Institute of Medical Research, and Michael Alpers, that has worked in this area for many decades. With the population geneticists, I work with Garrett Hellenthal at UCL and Ida Moltke in Denmark. And the work I talked about at the end was work of PhD student Liam Quinn, who's moved and married a woman from Denmark. Thank you so much for your attention. Sorry to go over. I don't know if there's time for questions, but yeah, super. Thank you. Has that been too baffling, too scientific? Yeah.
Sorry, excellent talk. Thank you so much. Thank you very much. Fascinating talk.
Thank you.
A really quick question, probably maybe a stupid one. You said that you're collecting, sporadic cases of CJD. How do you know that it's sporadic before it's basically terminated?
Yeah, that's a good point. Yeah, we make an assumption based on the clinical presentation, the absence of a family history of the disease, and we go on to confirm that genetically. But you're right, in some instances, probably less than 1%, we're wrong and we make a mistake and we find a mutation and it turns out it's genetic. And those are excluded then from further study. And the family is informed, But so that's really how it works. It's kind of a little bit in retrospect, in fact, and occasionally we do make a mistake, yeah.
Hi, Simon. I'm just wondering whether that protective codon 127 polymorphism is protective against familial forms of disease, if it would stop misfolding of a protein with the familial mutation.
That's a good question. I don't think we've done that. There aren't great models. There aren't, highly penetrant early onset models of human inherited prion disease. That would be an optimal kind of experiment to cross, a model like that with 127V and see whether it's protective. That would be a nice thing to do. They're only recently being developed when they tend to get kind of, Tom there is more of an expert on this than me, but the models that have been developed tend to develop disease later in a mouse life and sometimes patchily later in life. So it's not easy to look for modification. It's a very long-term experiment and it would need a large number of animals to be well-powered, but yeah, it would be a nice thing to know. Yeah, I agree.
Thank you for that talk. Just wondering if you had any thoughts on any implications from RT-QuIC being positive so many years before symptom onset, as that would relate to transmission risk or infection control issues?
Yeah, that's a good question. I don't think so, no, because I think we've got reasonable evidence that the vast concentration, or vast amount of prions made in the body are held within the central nervous system of those subtypes of disease, and so that diminishes the health risks, I think. It is legitimate to be concerned about possible transmission from people that carry inherited prion disease mutations, but I think that's a theoretical risk that hasn't been proven yet to actually exist in real life. I'm not aware that there's been any iatrogenic transmissions from somebody healthy, pre-symptomatic. So I don't see there's anything new in these data that change that, the fact that we can detect prions early on. I think it's a surprise for me that we can detect them for so long, seemingly doing nothing to the brain. There's no signal of neurodegeneration during these years in most of the people that we've studied, so they seem to be silently replicating prions for years, and then something must change. There must be some switch that leads then to a very rapid neurodegenerative disease. I think that's fascinating, and I don't understand it. But I note that it's not dissimilar from what has been observed in mouse models too, with a long pre-symptomatic period of high prion concentration prior to neurodegeneration. But it's obviously, it seems to me attractive to ask the question of whether therapeutic intervention in that stage will be meaningful. Sonia. Hi, Sonia.
Hey, Simon. I've been thinking about this E200K RT-Quick finding, and as you know, it's something we've seen too, and thinking about how E200K specific it is, given the current limitations of RT-Quick.
Yeah.
And what I would love to see somebody do is throw themselves into optimizing RT-Quick for other genetic subtypes.
Sure.
So I just thought I'd say that,
I
and see
agree.
if maybe you want to do it.
Tell my grant reviewers, Sonia, that, and I mean, Dr. Mock did work on that for his PhD. It's not currently funded, and I completely agree.
Yeah.
If we had an RT-Quick test that worked for all the different mutations.
Because you have to ask whether the kinetics are genuinely different, or
Sure.
whether our ability to see things is just different, and I think we have no idea.
Sure. No, absolutely. I mean, a good place to start is seed amplification assay, for want of a better term, that works from brain tissue of deceased people with the other mutations. We don't have that really yet, do we? And that is a big gap. Yeah.
I wonder if you or others would comment on the possibility that we're not detecting but creating prions with RT-Quick and the E200K, and that could it be that the nascent E200K, which is much more susceptible to misfolding, is just reacting more quickly in the mixture, and we're not?
Sure.
Yeah.
No, that's a reasonable point. And I think that question, so, if I understand what you're asking, is it just a false signal related to the fact that you've got a mutant protein there that you're putting into an RT-Quick reaction? And I think the answer to that question comes from clinical observations. So in other words, is the finding of a positive RT-Quick meaningful? And I didn't know at first, but I'm convincing myself it is, because I made reference to this in the presentation. We haven't had a conversion of 200K without a positive RT-Quick. And I think whilst we're not feeding back this information to individuals in the study, the study workers, we are aware of people that are RT-Quick positive, and we're making mental predictions of who may develop the disease next, and it's proving to be correct. And I think that means that it is more than just simply a mutant protein that is triggering a misleading or a meaningless RT-Quick reaction. But in order to answer that definitively, I think, as we were discussing the other day, Michael, I think it would be helpful, to gather together the world's experience like this, because I know, Sonia, you've had a number of similar stories from your cohort, and I think our Israeli friends have as well. And one thing we could do is look and say, well, from the point of the first positive RT-Quick test, what's a kind of survival curve for how, for how long you remain disease-free, and compare that to somebody with a negative RT-Quick. And I suspect those curves would look already, based on the world experience, very, very different. Yeah.
Great, thank you. On that same note, can you titer your RT-Quick?
Yes.
And if you're finding an increase as
No, it just seems
it's
to be flat.
flatlined?
Yeah, yeah, just flat. Yeah. Although we've only done a small number. Yeah.
This may be an impossible question to answer, but with respect to the genetic risk factors that you're trying to identify, is there any epidemiological evidence for genetic transmission among people with these higher risk factors?
Yeah, they're not such strong risk factors. So, you know, the kind of risk factors that we're detecting are nothing like, you know, like say you carry an E200K mutation, maybe that increases your risk of developing CJD by age 70 by, I don't know, let's say 2,000 or 3,000 times. But the risk factors I've been talking about are much, much weaker than that, maybe alter risk by 10, 20%, something like that. So a completely different ballgame altogether. They're subtly modifying it. I think, I say you've got a world expert on this subject sitting in the front row here, Eric, who's proven really nicely by looking across lots of different diseases and different genes that actually this doesn't mean it's irrelevant for drug development. You know, you've got an anchor there that's telling you that something, that if you've got a genetic risk, even if it subtly modifies the risk of a disease, it could still be very, very relevant to drug development. And I think the main reason for that is the genetic variants that we're detecting maybe only just very subtly alter the expression of that gene. They maybe only change that gene's expression by 10 or 15%. Maybe a drug could do an awful lot more than that, or it could be much more specific in terms of how it interferes with it. So it's definitely worth understanding why these hits that we've got work. They may give us fundamental insights into the disease, and you don't know where that's going to take you. You don't know what opportunities might come out of that. But the best way to exemplify that is with, Tom is going to speak, Tom Cunningham's going to speak later if you attend his talk on our follow-up of Syntaxin 6 and where we've got to with that. And I think we're really starting to get there and understanding how that works. And we are finding some much stronger effects in the mouse models in terms of risk when we take out the gene altogether. So not so deterministic, no, but still meaningful, I think, if that answers your question. Yeah.
Thank you.
Yeah. Thanks, you're really warming up now. This is
With your genetic association studies, are the variants in STX6 and are the other genes you've identified common enough that you could begin to look at the question of whether these same genes modify, say, the age of onset of an inherited prion disease or the incubation period of an acquired prion disease?
Yeah. I mean, we don't have such great numbers, but where we have looked, we haven't seen that modification. But there is, I'd hope to present a study today on exactly that question, the age of onset of inherited prion disease and whether genetically that can be modified. But I'm afraid I didn't manage to finish it in time. But that question is on the table. So we have put together around 1,200 samples from researchers worldwide, patients with inherited prion disease that died of a whole variety of different ages, people that had predictive gene mutations that never got the disease, we don't know why they never got it, but lived to an old age. And we're studying those genetically to try and answer that question exactly. Now, if STX6, GAL3SG1 comes up, that will be fascinating, won't it? But maybe there are completely different genes that alter those phenotypes. Yeah.
Thank you, Dr. Mead.
Yeah,
I'm going to try to ask this in a logical way, but in your study, your longitudinal study of genetic patients, you said that you see a positive test or positive presence of the protein with the RT-Quick test several years before onset of symptoms. Can I infer from that it's progressively a buildup or an accumulation of larger and larger amounts of protein, or just the evidence of?
Yeah, unfortunately, we don't know that. We don't know the answer to that question, which is, I think, a great question. Remember, of course, you don't get brain tissue. And it's, people have been really generous to running the study, but I think asking for a brain tissue sample would be would have a big impact on the success of the study, wouldn't it? So we get spinal fluid. which is a fluid which bathes the brain and communicates with the brain, but you have to make inference about what that means. As Valerie asked, we have looked at the concentration of prion seeds that we found in that spinal fluid over time, and we don't see a progressive increase. But I wouldn't want to assume that that means there isn't the buildup of something in the brain over that time, and which crosses a certain level that flicks a switch to develop the disease. But neither do I want to make the assumption that that is what's going on. We just don't know. And I think it's a fascinating subject about how it is that prions can replicate to high levels enough to be able to detect in the spinal fluid, but seemingly cause no harm for many years, and then something happens and somebody dies within a few months. I find that intriguing from a clinical science point of view, that there's some really powerful mechanism there we don't understand, and we need to. Thanks so much. Thanks for all your questions. Yeah, thank you.